Assalamualaikum, I am Ibtisam Ipangandaman, and for today's video, I will be discussing to you the metabolomics, the topic that I choose in our video presentation. So, what is metabolomics? Metabolomics is the large-scale study of small molecules, commonly known as metabolites, within cells, biofluids, tissues, or organisms. Collectively, these small molecules and their interactions within a biological system are known as metabolome. So in figure number 1, we have here the gene, mRNA, protein, metabolite. So just as genomics is the study of DNA and genetic information within a cell, and the transcriptomics is the study of RNA and differences in mRNA expression, and metabolomics is the study of substrates and products of metabolism which are influenced by both genetic and environmental factors. Metabolomics is a powerful approach because metabolites and their concentrations, unlike other omics, measures directly reflect the underlying biochemical activity and state of the cells or tissues. Thus, metabolomics best represents the molecular phenotype. So, what are small molecules? So, what are small molecules? A small molecule or metabolite is a low molecular weight organic compound typically involved in a biological process as a substrate or product. Metabolomics usually studies small molecules within a mass range of 50 to 1,500 daltons. In figure number 2, we have here some examples of molecules: glucose, stearic acid, cholesterol, and lysine. Examples of small molecules can be seen in figure 2 and include sugars, lipids, amino acids, fatty acids, phenolic compounds, and alkaloids. There is a great deal of variation in metabolites between species. It is estimated there are around 200,000 metabolites across the plant kingdom and somewhere between 7,000 and 15,000 within an individual plant species. By contrast, in humans, there are thought to be around 3,000 indigenous or common metabolites. These estimates are approximations that are likely to be underestimates because it is difficult to detect low abundance molecules. Nonetheless, it can be concluded that plants are particularly biologically rich by comparison with many other species. They also typically contain larger numbers of genes than other eukaryotes. The metabolome. The metabolome is the complete set of metabolites within a cell, tissue, or biological sample at any given time point. The metabolome is inherently very dynamic. Small molecules are continuously absorbed, synthesized, degraded, and interact with other molecules, both within and between biological system and with the environment. Metabolic reactions. Metabolic pathways are essentially a series of chemical reactions catalyzed by enzymes whereby the product of one reaction become the substrate for the next reaction. These reactions can be divided into anabolic or catabolic. The importance of metabolomics. The importance of metabolomics is the importance of metabolomics. Why is metabolomics important? The non-invasive nature of metabolomics and its close link to the phenotype make it it an ideal tool for the pharmaceutical, for preventive healthcare, and agricultural industries, among others. Biomarker discovery and drug safety screens are two examples where metabolomics has already enabled informed decision making. In the future, with the availability of personalized metabolomics, we will potentially be able to track trends of our own metabolome for personalized drugs and improved treatment. Strategize. Personalized treatment is likely to be more effective than our current medical population based approaches. How is metabolomics used? We benefit from metabolomics on various levels, from products and stress testing in food industries, control of pesticides, and identification of potentially harmful bacterial strains, to research in agriculture, crop protection and engineering, medical diagnostics in healthcare, and future applications in personalized medicine resulting in personalized treatment strategies. Applications of metabolomics. We have agricultural, biomarker discovery, and personalized medicine. In agricultural, the development of new pesticide is in agricultural, the development of new pesticide is a critical to meet the growing demands on farming. Metabolomics enables us to improve genetically modified plants and helps us estimate associated risk by allowing us to get a glimpse of their complete biochemistry via formative snapshots acquired at a different time points during plant development. Plant metabolomics is particularly interesting because of the range and functions of primary and secondary metabolites in plants. About 300 distinct metabolites could be routinely identified per sample a decade ago, and the number is gradually increasing over time. Biomarker discovery. Biomarker discovery is another area where metabolomics informs decision making. Biomarkers are objective indications of medical state observed from the outside the patient, which can be measured accurately and reproducibly. Biomarker discovery. Biomarker discovery is another area where the metabolomics informs decision making. Biomarker, Biomarkers are objective indications of medical state observed from outside the patient, which can be measured accurately and reproducibly. In metabolomics, biomarkers are small molecules or metabolites that can be used to distinguish two groups of samples, typically a disease and control group. For example, a metabolite reliably present a disease samples but not in a healthy individuals would be classed as a biomarker. Samples of urine, saliva, bile, or seminal fluid contain highly informative metabolites can be readily analyzed through metabolomics, fingerprinting, and profiling for the purpose of biomarker discovery. And the last one, we have personalized medicine. Personalized medicine is the ultimate customization of healthcare, requires metabolomics for quick medical diagnosis to identify disease in healthcare. We currently use classical biochemical tests to measure individual metabolite concentrations to identify disease, such as the blood glucose level in the case of diabetes. Metabolomics offers the potential for the rapid identification of hundreds of metabolites, enabling us to identify these disease states much earlier. So, designing a metabolomic study is we have the two main approaches that can be used in metabolomics are untargeted and the targeted approaches. The approach chosen will determine how you design your experiment, prepare your samples, and what analytical techniques you use. In untargeted or global approach, this method measures as many metabolites as possible from range of biological samples without any intended bias. In targeted approach naman, this method is used when you want to measure sets of metabolites and have specific biochemical questions that you want to answer. This approach is often used in pharmacokinetic studies of drug metabolism and when looking at the effect of therapeutics or genetic modifications on a specific enzyme. Metabolomics in health and disease. Metabolomics has several applications in health and disease, including precision, personalized medicine, single-cell, epidemiologic, population studies, metabolic phenotyping, and metabolome-wide association studies. Precision metabolomics and in combination with other omics disciplines as integrative omics. Single-cell metabolomics and single-cell lipidomics technologies allow high-dimensional characterizations of individual cells. Disease, heterogeneity, and complexity, identification, expression, and abundance of disease-associated small molecules, metabolites, and others, and live single-cell mass spectrometry, imaging MS analysis of human breast cancer samples at a single-cell level revealed cell-to-cell interaction and tumor heterogeneity, clinical biomarkers, and different metabotypes of disease severity correlated to exposures and biological outcomes have been studied and identified through metabolomics profiling. Metabolome-wide association studies and metabolomics fingerprinting and footprinting techniques in individual and population which will enable precision and medicine and public health care. Studies moving from genome-wide association studies to metabolome-wide association studies were first described in 2008 as environmental and genomic influences to investigate the connections between phenotype variation and disease risk factors. Rattray and colleagues suggested that in conclusion, MS-based metabolomics or lipidomics provides a useful approach for both identification of disease-related metabolites in biofluids or tissue, and also encompasses classification or characterization of disease or treatment associated in molecular patterns generated from metabolites. In summary, metabolomics is the large scale of a small molecule or metabolites within cells, tissues, or organisms. Application of metabolomics are found within the pharmaceutical, healthcare, and agricultural industries, among others. There are two main approaches used in metabolomic studies. We have untargeted global or targeted species. The untar- there are two main approaches used in metabolomic studies. We have untargeted or global approach and targeted or specific approach.